question we are attempting to answer is, why does a high-fat diet decrease skeletal muscle GLUT4 content? Our hypothesis is that high-fat feeding increases ceramides, a reactive lipid species in skeletal muscle cells. Ceramides inhibit the translocation of GLUT4 to the muscle cell membrane by interfering with AKT phosphorylation in the insulin signaling cascade.
Our match to this problem is membrane GLUT4 content. Our inflow is GLUT4 vesicle translocation from the cytosol to the cell membrane, and our outflow is movement of GLUT4 back to the cytosol. However, for this problem, we will be focusing on inflow and will not be addressing outflow any further. GLUT4 vesicle translocation to the cell membrane occurs by insulin signaling, whereby insulin binds to the insulin receptor, which activates insulin receptor substrate. This in turn activates PI3K, which both activates RAC as well as AKT. AKT inactivates AS160, releasing GLUT4 vesicles from the cytosol to be transported to the membrane via RAC, and this increases GLUT4 membrane content. Consumption of a high-fat diet, intake of exogenous fats increases. Via digestion, this increases plasma-free fatty acid concentrations, and via CD36, this is taken up by the cell to increase fatty acid acyl-CoA concentrations. This leads to an accumulation of both DAGs and ceramides, which are reactive lipid species. DAGs inhibit insulin receptor substrate, and ceramides, which is the focus of this presentation, inhibit AKT. When AKT is inhibited, it decreases its inhibition of AS160, and therefore there is more AS160 inhibition of GLUT4 vesicle translocation, leading to a decrease in membrane GLUT4 content.
To test our hypothesis, we looked at a journal article from the Journal of Molecular and Cellular Biology called Regulation of Insulin-Stimulated Glucose Transport, GLUT4 Translocation, and AKT Kinase Activity by Ceramides. The purpose of this article was to investigate whether a short-chain ceramide analog, C2-ceramide, inhibits glucose uptake, GLUT4 translocation, and AKT phosphorylation and activation in 323-L1 adipocytes independently of any effect on IRS1. The methods for measuring glucose uptake in plasma membrane GLUT4 were assays. Western blots of total cell lysates were prepared and analyzed. AKT kinase was analyzed through assays, and IRS1 was immunoprecipitated by solubilizing cells. C2-ceramide disperses more easily in the incubation medium and is metabolized more slowly than ceramide, and it is thus a reagent commonly used for investigating ceramide's effects. The results of the study showed that C2-ceramide inhibited insulin-stimulated glucose uptake into 3T2-L1 adipocytes without affecting basal transport rates. C2-ceramide inhibits GLUT4 and IRAP translocation as well. C2-ceramide markedly inhibited the phosphorylation, mobility shift, and precipitation of AKT kinase activity. C2-ceramide's effects on AKT were apparent within 1 hour of treatment. C2-ceramide did not inhibit insulin-stimulated phosphorylation of AMPK, PP70 S6 kinase, or the ribosomal S6 subunit. Moreover, C2-ceramide did not alter the insulin-induced mobility shift observed for IRS1 or PP70. These results indicated that C2-ceramide specifically inhibits insulin stimulation of AKT without affecting other insulin-mediated pathways. C2-ceramide does not inhibit tyrosine phosphorylation of IRS1 and does not inhibit recruitment or activation of PI3 kinase. In conclusion, C2-ceramide antagonizes insulin-stimulated glucose uptake and GLUT4 and IRAP translocation in 3T3-L1 adipocytes as it inhibits insulin-stimulated AKT phosphorylation.